This talk is about finding cross-species orthologs with local topology, and as it turns out, sheaf theory as well. This talk is joint work with a number of people and organizations with SRC Inc. Chris Caprero is our programmer, the Applied Topology team at PNNL, and Baylor College of Medicine for the data and post-processing, and I'd like to acknowledge Reza Ghanandan of the DARPA DSO Simplex program. So the idea is, can we identify related proteins across different species of organism? So if you have two species, say species A and species B, they each have a variety of proteins. Those proteins have certain functions, they interact with each other, and they're also genetically related. Proteins within an organism, of course, do interact, and we have some knowledge of their interaction network. And based on the protein sequences, we can group proteins together across species and within a species into clusters of orthologous groups. They're sets of genetically related proteins. And in essence, this talk tells you a method for finding those COGs. The data set we're going to talk about is StringDB version 9.1. The data that was given to us from Angela Wilkins and Daniel Konecki from Baylor College of Medicine. They picked out 7 species that were interesting. They pulled out a bunch of protein-protein interactions and a list of COGs that are known. In all, just under 60,000 proteins are represented in the data we're going to process. So looking at the data, here's kind of the question rephrased a little bit. On the left, we have a protein-protein network from human and an analogous one from mouse. We happen to know the two proteins in red are related. That's how we kind of found this. And you can see the protein networks look very similar. Not exactly the same, but quite similar. And so the question is, can we identify, without looking at the sequences, that in fact these two proteins in red ought to be related? The reason for doing this is because finding the related COGs, finding the proteins that are genetically related by sequence, it works. It's a very reliable method, but it's very, very slow. You have to sift through fairly long sequences, and there's a lot of processing involved. If you could just work from the networks, that would be a lot sparser data and would be quite nice in terms of having a good functional relation as well. So that's the goal. And one of the first things we noticed was that if you look at these two proteins, their networks do look very similar. And if two proteins are in the same COG, they tend to be in other COGs together also. So if you look at those same two proteins that I had circled on the previous slide, the human one and the mouse one, you can see that they share many, many COGs together. As in, not only are they have similar protein-protein interactions with their respective proteins in their organisms, but their proteins are related, and their neighbors are kind of related. There's actually only 2 differences between the lists of COGs that these two proteins are in. So that's a very strong indication that these proteins should be related and can be inferred without looking at the sequence data. This is somehow boiled out from the sequence data. So the key insight is that if two proteins have similar interaction structure with neighboring proteins within that organism, and their neighbors are related, as in they're in the similar set of COGs, then those two proteins probably ought to be in the same COG as well. And here's how we're going to cast this into a topological processing method. We're going to build a sheaf over a base space. The base space is going to be the interaction structure within a species. So the goal is to narrow down the search space to check to see if you've got orthologs. And the tool we're going to do is we're going to do something that amounts to a particular kind of graph alignment. But that's not all. Once we kind of have narrowed in on proteins that have similar interaction structures within their respective organisms, then we're going to make use of what COG structure we know to kind of leverage out and zero in on the groups of proteins whose sequences are related not only to each other but across species. And the tool we're going to use is something that's relatively new that our group has discovered, a mathematical tool called the consistency radius, associated with sheaf of pseudometric spaces. And it's a sheaf of pseudometric spaces and they're really very simplistic sheaf structures, you'll see. So what's new about this idea? Well, the usual procedure that people in bioinformatics tend to do is they tend to focus on the sequences. They take the sequence data, they've got partial protein interactions to kind of inform which proteins they're going to look at. No COGs information to start with, and they produce the COG network purely from that sequence data. Our procedure kind of is a bootstrapping. We make use of the protein interactions as a pretty important part. It forms the base space of our sheaf. We take the partial COG network, as in what is known about the COGs, but we recognize that they're incomplete. But we take no sequence data. And from that we try to complete, or if you will, do some link prediction to build out a larger COG network. As a flowchart, this is how the process runs. We take the two species' respective protein-protein interaction network graphs. That's our input on the upper left. We take those, we convert them into abstract simplicial complexes through the process of forming a flag complex. In some sense, this isn't any more or less information than what's in the graph, but it's interpreted a bit differently and a bit more topologically, or at least more, not per se more topologically, but more higher dimensional. We extract neighborhoods out of that and do essentially a neighborhood comparison. That's a local network alignment. Our colleagues have informed us that doing global network alignments are not necessarily likely to succeed because there's a lot of differences, not only between organisms, but just in terms of coverage of data. So we're really focusing on a local network alignment. Once we've done that, and we sort of threshold, we have a candidate set of protein pairs, then we zero in, and we're going to transition from processing the base space to processing a sheaf. And what we're going to do is we're going to build a sheaf over a slightly modified neighborhood in which we've added a new edge between the candidate protein pairs that we'd like to see whether or not they ought to be in the same COG. This is a joint protein local abstract simplicial complex. We sheafify over that, so we add some additional data from the COG database. We construct an assignment to that sheaf, so we fill out some known data, compute the approximate section radius or the consistency radius, threshold that, and based on that we have an understanding of whether or not we're looking at a pair of proteins that are really indeed related to one another or not. And when you look at this process, you can say, all right, well, how well do things run? Well, purely looking at the base space, about 75% of the protein pairs that we end up proposing as being related are already manifest in the COG database. That says that most of the time we agree with what's already known, and that's good. And because essentially what that's saying we're cross-validating against the sequencing. But there's about 25% that we're proposing at that stage that may or may not be new, and they're at least novel. After we're done doing the sheaf processing, about 85% of the data is manifest in the COG database. So we've rejected a number of proteins that look like they have similar network structure, but are not really related, or at least their neighbors aren't related. And that improves things. Now the remaining 13%, we propose as potentially new COGs, and we cross-validate them with this BLAST, which compares protein sequences. Somewhere around 30 to 50% of those are indeed true actual COGs that simply weren't manifest in the database. So what we're doing here is we're able to fill out that COG database with some degree of certainty. It's not perfect, and as you'll see, the accuracy, the likelihood that a protein pair is indeed a new COG is quite strongly related to the consistency radius associated to this sheaf. Okay, so let's dive in and see how that works. So the first thing we're going to do is we're going to take the protein-protein interaction network graph. It starts out as a graph. The vertices are proteins, the edges are interactions. And what we do is every time we see a click, an edge between every pair of vertices, those become simplices. So we call that process the process of forming a flag complex or a flagification. The payoff of doing this is that it's sort of better representation for multi-way interactions between proteins. If you see large simplices, that kind of indicates to you that you're dealing perhaps with a bunch of proteins that really ought to fit together as a functional unit. This is intuition only, though. And in some sense, the flag complex carries no really more information than the graph itself. Now, if you try to do a local network alignment or local abstract simplicial complex alignment, there's a variety of tools you might try to do that. There's several different local graph metrics that you could try. So you can look at a vertex degree histogram and see how well those vertex degree histograms agree as functions. You could look at various kinds of spectra. You could look at some graph density or some graph Betty numbers. So some of these are topological, some of these are not. It's worth noting, as an aside, that homology and spectra are very tightly related through Hodge theory. In a graph, the graph Laplacian determines homology, and it's convenient, it's widely used. The graph Laplacian contains some geometric information, and it determines the topology through the Hodge theorem. Well, that's quite convenient, and lots of people like that, and lots of people in graph theory will use the graph Laplacian. Very easy to compute, makes a lot of sense. For abstract simplicial complexes, there are, of course, higher dimensional analogs, higher Laplacians, and they tell you more about the other homology classes. Hey, that's cool. And there's some additional geometric information. The higher Laplacians definitely determine homology, but for whatever reason, they're not very much used in data science. Frankly, I'm not sure why, and I'd like to put out a bid to say, hey, why not? Maybe they're good. Maybe you should use them. Now, I have to admit, we're not computing them either in this particular case, although it's not that they're really hard to compute. Okay, and the aside. Coming back to those metrics, some of them are topological, as you can see, and you can see the correlation between known COGs and matched networks is reasonably high for some of them. In fact, for the topological ones, purely topological ones, the correlation is pretty high. Now, what that's saying is it's saying local topology of the networks being similar is an indication that two proteins are probably in the same COG, but it's not conclusive. Remember, in our particular excerpt that we're looking at, it's on the order of 60,000, a bit more than 50,000 proteins, and the full StringDB database is 10 times more than that. So if you've got an error of 10%, that's still a lot of proteins. So what we want to do now is we want to use this merely to reduce the search space and then drill down using a sheaf over this abstract simplicial complex to give substantially more accurate results. So the mantra in local topology, or algebraic topology, is local to global. The problem with this is that it tends to exhibit poor scaling. Everyone in applied topology is always fighting this to some extent, usually computing homology, which is the standard global invariant that people like to use. It's cubic in the number of simplices. It requires a lot of linear algebra. Usually that's good. Linear algebra is easy to compute, but it's not always good. And indeed, if you're dealing with COGs, they're kind of more semantic. And the real data usually can't be globalized if you try to do a local to global transformation due to errors. So maybe that's not such a good idea. On the other hand, very little effort's been expended by researchers in this field about partially global results, sort of local sections, and various kinds of locality. We've been looking at them recently. We found that there's some interesting combinatorics present there. That's not the subject of this talk, although if you're interested, I strongly suggest you go have a look at Emily Purvine's work. But the payoff is that partially global results are more realistic, they're a little more tolerant to noise, and they're a lot easier to compute, not dealing with cubic scaling and that sort of thing. I've realized there's worse scaling than cubic, but cubic scaling is definitely nothing to sneeze at. So the idea is we're going to make use of this sort of sheaf theoretic approach to modulate our local analyses. So we start with our abstract simplicial complex. It consists of simplices, various tuples of vertices. Remember, each one of these simplices is a list of proteins that are interacting within an organism. Well, you could view this equivalently as an attachment diagram. It tells you how the simplices fit together. It's a partial order going from small sets to larger sets. A sheaf over this sort of thing is a set assigned to each simplex. Each set is called a stalk, and this is what I'm building out in this example here is a sheaf of vector spaces on a simplicial complex. It's worth noting that those prepositions are helpful there. Sheaf is two things. It's of and on, of a certain data type on a certain data type. We will be focusing with, as it happens, sheaves of sets on simplicial complexes in our algorithm. And it's not just the stalks, it's also the restriction maps. Each one of those relations between faces, or face inclusions, is given a restriction map, and that restriction map has to be chosen such that the diagram commutes. So all of those restriction maps have to be such that every time I walk around the diagram, if I compose maps as I go, that needs to be path independent. Okay, so if I assign data, if I play values at the vertices, and this corresponds to protein metadata, for instance, the COGs, the question is there consistency? If I can come into one of these red question marks from two different arrows, do I get the same answer following along those restriction maps? Sometimes I do, sometimes I don't. Again, being realistic, I don't usually get the whole thing. I'm going to get something partial like this. So I end up with a situation where in some places, say in the upper right there, I get consistency. I get the vector 2, 0 coming from both the top and from the right vertex. But in other cases, I don't. So if I go from the top down or the bottom up, I don't get the same answer. And everywhere I don't get the same answer, I've put the red X. So I want to measure the amount of inconsistency, and this tells me somehow how consistent the data are with the base space that I've built. Okay, well, one recent useful theorem that one of our PNNL colleagues, Brenda Bacastis, proved, and she's got a paper up on Archive if you're interested in reading about it, is that we can compute the cover, the largest maximal cover by largest cover by local sections in an algorithmic way. So that's kind of handy, and that gives you some notion, by studying that cover, some notion of the quality of the assignment of data. Well, that's pretty neat. There's another more numerical way that you can get at it, called the consistency radius, and that plays an important role in our algorithm here, where essentially what you do is every time you have multiple ways of coming into a face, you ask, what's the radius of all of the set of values that are present there? Where you have exact consistency, as in, again, to the upper right, that vector 2, 0, the distance there, the radius is 0, because both values are 2, 0, and the radius is 0 then. But if you say come from the top down or the bottom up, so if you multiply the by the 1, 0 there, or the 2 by 3 matrix, you end up with a vector 0, 2 coming from the top, and a different vector is 0, 1 coming from the bottom, and those are distance 1 apart. Of course, in the middle of the triangle, where I have 3 arrows coming in, I have a whole bunch of values to check. Because the diagram commutes, I only have 3 values to check, 1 coming from each of the vertices. So if you maximize that particular radius over all the faces, we call that the consistency radius. If you're interested, you can read the reference at the bottom of the slide, where this is defined carefully and a bunch of useful properties about it are proven. Okay, we're going to use that. And what we're going to do is we're going to come back to our protein-protein interaction networks. In our algorithm, we actually use the 2-hop network. I'm showing you only the 1-hop network. Nevertheless, so I have 2 species sitting here, and I've got a bunch of proteins. Proteins A, B, C, D for species 1, and proteins 1, 2, 3 for species 2. And for whatever reason, I've zeroed my attention in on protein A and protein 1. I suspect that they're related. I'd like to know. So what our algorithm does is it says, okay, those 2 proteins are the ones that I'm interested in. I'm going to add an edge between those proposed orthologs, those proteins that I believe to be related. And now what I'm going to do is I'm going to build a sheaf over this larger abstract simplicial complex and ask, what is the consistency radius over it? If the consistency radius is small, I'm going to say, I think these 2 proteins are related. If not, I'm going to say, probably putting that edge in there wasn't such a good idea. Okay, so how does this work? Well, if I'm going to build a sheaf, it's got to be something of this form. It's got to have this kind of diagram. So the sheaf has to have a bunch of stalks and a bunch of restrictions, and it has to be of this shape. But I need to tell you now what those are. So what I'm going to do is I'm going to fill out more or less the most obvious way that comes to mind is I'm going to list all the possible COGs that might be there and all the possible sets of COGs that might be at every stalk. So at every protein, the possible COGs that are in play. I'm going to let all the restriction maps be identity functions. We could use something more sophisticated, but this appears to work algorithmically. Okay, but now I need to tell you what an assignment is. It would seem like an assignment should be to tell me the set of COGs that each protein participates in. However, when we look at the data, this doesn't exhibit much self-consistency at all, as in even within a single species. Well, that's not very good. But if you remember at the beginning of this talk, I said that within a protein, or within a species, the COGs that pairs of related proteins are in tend to be pretty overlapping when you include their neighbors. So if we do that, that improves the consistency quite a bit. And you can see that just from this particular diagram here. If I start out here that the protein on the very left has KL, and the one at the top has empty sets not in any known COGs, and the one that's sort of at the very bottom of the diagram, the L, is only in that one COG. If I now go through and expand that include the list of all of the COGs of themselves and their neighbors, now it's actually a lot more consistent. In fact, in species 2, you can see there's complete consistency throughout. Now, I don't have a pseudometric on hand yet, but I can certainly pull a set pseudometric off the shelf. And I can certainly compare whether or not I have consistency. Here's a maximal local section. If I don't have a global section, then that pseudometric is quite useful. Here's, for instance, one possible set metric that I could use. There are plenty of others. I could use Chicard index, for instance. I could use lots of others. This particular one, if I happen to use it, what I've marked in green are the radii of the sets of values present at each face. And you can see that the resulting consistency radius right by inspection here is 0.18. And so essentially what I do then is I threshold that 0.18 and I say, all right, that I'm going to assign and associate to that pair of proteins. So it carries some information that's a little bit more than just those pair of proteins, because it also carries around the consistency within the rest of the network. And in principle, one could modulate that, but we're not at the moment. Now, we can go ahead and process all this data, and we end up with a table that looks like, for instance, the one that's in that spreadsheet there. And so the way this works is we propose protein pairs, and we propose which COGs they ought to be in. And that metric that's listed there, that column D, is our consistency radius. So going ahead and doing this produces a list of proposed COGs. That's the prediction stage. If we want to validate this to see how well this works, we run a process that's pretty standard in bioinformatics called Reciprocal BLAST. We take the sequence from the first protein, and we use BLAST to find all the potential homologs in species 2. And then we take each of those and blast them back into species 1. And we ask, how well do we recover the same protein we started with? So does each protein that's present, or each proposed protein, pick the neighbor the COG we predict? And does that one predict the first one? So if each of these proteins pick each other as being related to one another, then we say, yep, that's a match. And so we essentially ask for this to happen either in the top first one, as in you pick your best friend and your best friend picks you, or at least you got to be in the top n of each. Okay, so that's the validation process. As I said, it's a pretty well-established process, and it's a pretty rigorous process. It is entirely sequence-driven. There's no protein network structure here, and there's no actual knowledge of the COGs either in the validation. So what we're doing is we're validating our process, which is sort of bootstrapping this sequence information that's embodied by the COGs, to find more, and then we're validating. When we do this is, if you will, the payoff slide. You can take a look at the consistency radius, and notice as the consistency radius increases, you get less and less consistency. The proteins are indeed less related. The percent of proteins that end up with similar sequences, those that are these reciprocal best matches, drops off rather precipitously. So if you're looking at the red curve, those are the top hits. I think this protein is my best match, and it thinks that I'm its best match. So you can see of the proteins we proposed, which, remember, there's about 87% of the ones we propose are already in the database. So this is of those remaining ones, the new ones, about 30% of them. Yep, those are top hits. It peaks up, if you look within the top 3, about 50%. So what this says is that more similar network topology and COG label structure definitely indicates that the proteins are going to have much more similar sequence. And as the topology it becomes less similar, as the sheaf structure becomes, the data assigned within the sheaf becomes less similar, the proteins are less likely to be related. So the conclusion of this is that consistency radius really is a measure of relatedness of protein pairs. 30 to 50% of our most likely protein pairs are truly novel orthologs, and that protein interaction network and COG self-consistency together, pull them together, you can predict sequence similarity. That's a pretty powerful idea. So this network structure is very supremely useful. In fact, there's a sort of a speculation that this is because important functional networks are preserved through evolution. And it's entirely possible that some of our protein pairs that don't have similar sequences are actually playing functionally similar roles. There may be convergent evolution. We don't know at this point. This is still very new. And it's entirely possible that they may play similar roles in different biochemical pathways.
So, of course, we're now continuing some further validation. We'd like to finish processing all 7 species. I've just shown you species for human and mouse. Do some retrospective analyses. StringDB 9.1 is a year out of date. Maybe we can predict what was later studied, later found. Can we predict what has been discovered over the past year? That would be kind of a neat study. And I would say sheaves seem to be a natural way to transfer information about model organisms, say mice, to humans, but we don't actually know if that's actually effective. So we'd like to extend our processing beyond the kind of metadata that we've looked at, for instance, drug interactions, diseases, or pathway networks. And for instance, we're now working on the biopsych repository to see if we can make use of that structure as well. So thanks for having a look at this. Hope you enjoyed it. And if you've got questions, let me know. Thanks a lot.